Good evening, class. We would like to look at the structure of of antibodies, and also the functions of antibodies. We'd like to look at the structure and functions of antibodies. So let's go. Okay, so we have, as part of our objectives, we'll be looking at the major classes and functions of immunoglobulins. We'll also look at the structure of immunoglobulin, antigens, immunogens, vaccines and the types and functions of vaccines. But we may not touch on the vaccines so much because we've already done it in the active and passive immunity. Okay, so let's watch a video. Let's watch a short video on antigens, on antibodies.
The antibody is generally consisted of the heavy chain and the light chain. The heavy chain is always the constant region, whereas light chains are composed of both constant and variable regions. The antigen binding sites are present in the light region, whereas the Fc region, which binds to immune cells receptors, are present in the heavy chain region. The antibody can be a single molecule, such as IgD, IgE, and IgG. Or they can be a couple of molecules connected together, such as IgA antibody, or a cluster of 5 antibody molecules connected together, such as IgM antibody. Now we will have examples of antibody functions. Firstly, agglutination. Here, the antibodies form a large complex with bacteria. This complex neutralizes the bacteria and facilitates their removal by phagocytic cells. Secondly, antibody-mediated phagocytosis. The binding of antigens to the antibody activates the complement system linked to the antibodies. The complement system in turn works as a navigator for the phagocytic cells to the site of the antigen-antibody complex. Complement proteins activate the phagocytes, which become eager to engulf the antigen-antibody complex and remove them from the blood. Thirdly, antibody-dependent cellular toxicity, where antibodies attack antigens on the surfaces of the targeted cells. Then, the Fc part of the antibodies binds to specific receptors on the natural killer cells. The natural killers do not phagocytose the antigen-antibody complex, but instead they release a group of perforating enzymes which ultimately destroy the target cells. This video is narrated by
first time you've heard about antibodies, you may not know how important they are. Antibodies are one of the warriors inside our body, helping us fight off nasty invading particles. They are a type of protein and a key component of our immune system. If a person doesn't produce or overproduces antibodies in the body, the person will become very ill. So where do antibodies come from? Antibodies circulate in the bloodstream and can appear anywhere throughout the body. Their function is to recognize bad agents called antigens and then trigger further chemical reactions in the body to remove the antigens. Antigens are foreign substances that are capable of triggering an immune response. They can be viruses, fungi, bacteria, and some non-living substances such as toxins, chemicals, and foreign particles. They can even be allergens like dust, pollen, or certain foods that cause allergic reactions. Antibodies are produced in the immune system in response to the presence of an antigen. So what do antibodies look like? Antibodies are also known as immunoglobulins. They are generally a Y-shaped protein consisting of 4 polypeptides, 2 identical light chains, and 2 identical heavy chains. The terms light and heavy simply mean larger and smaller. The 4 polypeptide chains are connected by disulfide bridges. Here, at the tip of each arm are the antigen binding sites. These are really important parts for attacking invading antigens. Similar to the lock and key mechanism in enzymes, the shape of the antigen binding site is specific to the antigen it's targeting. If part of the antigen fits into the antigen binding site, the antibody recognizes the foreign antigen and binds it in. This stops the antigen from causing more damage elsewhere. 2 identical binding sites means any given antibody can bind simultaneously to 2 identical antigens. Millions of antibodies with slightly different tip structures exist, with each variant binding to a different antigen. This enormous diversity of antibodies allows the immune system to recognize an equally diverse range of antigens. By binding in an antigen, an antibody can stop the antigen from entering or damaging cells. This is known as neutralizing. The antibody can also tag the antigen for attack by other parts of the immune system. In mammals, there are 5 immunoglobulin isotopes. They perform director roles and help direct the appropriate immune response for each different type of foreign invader. Antibodies are cloned in laboratories. To discover how and why, watch this video.
Okay, so let's proceed on a more serious note. Okay, so antibodies are also referred to as immunoglobulins. So we use antibodies and immunoglobulins interchangeably. Antibodies are circulating proteins produced by vertebrates in response to an antigen, like what you saw in the video. Antibodies mediate humoral immunity. They were first discovered as proteins against diphtheria toxins, hence the name antitoxin. When realized that antibodies can be generated against different substances, the term antibody was introduced. The substances they bind to are called antigens. Okay, so that's it. So we have this is a B lymphocyte, B cell or B lymphocyte. It has antigens on the surface. This is a naive one. When this naive cell becomes activated, and the only thing it becomes activated is through antigen stimulation. When it becomes activated, activation is what? Plus what? Antigen. Activated is what? Antigen. When you present an organism to this B cell, what happens is that this B cell will now change to what? A plasma cell. And a plasma cell, it will now change to what? A plasma cell. A type of B cell that produces what? Serum or systemic antibodies. So the plasma cell will now produce what? Antibodies in circulation. Antibodies in circulation. This is the structure of antibody. Antibodies in circulation. Find difficult to draw. Antibodies in circulation. So plasma cells produce antibodies into the blood to fight infections. So the antibodies we have there, this is the site that binds the antigen. There are two of them. The site that binds the antigen. So we have the constant variable region. This is a variable region. This part is variable region, and the down part to this part. So the down part here, from here is the up where it binds to the antigen is called variable region because it can open its mouth. This one can open its mouth to accommodate the antigen. But from here down, then from here to here, from here to here, from here to here, they are called hot, heavy, constant regions. Okay, so we'll see that in our lecture. Okay, so let's see. So, still on the introduction. So antibodies and T-cell antigen receptors are major players in the adaptive immune system. Antibodies are synthesized by B cells and exist in two forms. We have the membrane-bound antibodies on the surface of B cell. That is this one. These ones are membrane-bound, plasma membrane-bound. Then we also have secretory antibodies in circulation. They can be secreted in circulation or found in mucous secretions or interstitial fluids. So it's not only in the blood that they are found in. So these ones can be in the mucosal lining of the stomach, mucus secretions. They can be found in mucus, in the interstitial fluids, interstitial fluids, and also in the blood. So these are circulating antibodies, and they are produced by our plasma cells. And it is the naive B cell that, upon antigenic stimulation, when you present an organism to it, it changes to become plasma cell, which is the active form of the B cell, which produces these systemic antibodies that are located in the mucous membranes, interstitial fluids, and your blood. So, at the effective stage, when there is antigen-antibody reaction, what happens is neutralization. The antigen is taken out of the blood or destroyed. Opsonization, meaning that the antibody will coat the organism, like what we saw in the complement. So if this is the organism, many antibodies will come and coat it. The antibodies will come and coat it like this. They will use their FC fragments to coat them. So they will come and coat. The fragment antigen binding site will coat. So this one, if you have a typical antigen antibody like this, this is what we call this one the fragment antigen binding, or FAB, and this one we call it fragment crystallizable, or FC. This one too is FC region, and this is fragment antigen binding. This one is also fragment antigen binding, or FAB. This is the side that binds the antigen, and the binding of the antigen neutralizes the organism. Then when the antigen is, this is the antigen, if this is the antigen, if you coat the antigen with many antibodies, we call this one opsonization. It means that you have coated, the woman has worn some lipstick and other things, want to attract the men, so the men will also come and coat it. Okay, so let's continue. We have phagocytosis, that's the swallowing of the organism. Then antibody-dependent cytotoxicity, antibody-mediated mast cells activation. This one we see that one in hypersensitivity reactions. That will be our last lecture. Okay, so introduction once again. Okay, so let's continue. So serum sample that contain detectable antibody molecules that bind to an antigen is commonly referred to as antiserum or antisera. So when you go to the laboratory and start working, they will bring you, some of your seniors will tell you that process this blood into a serum. So how do you process a blood into serum? What happens is that you centrifuge the blood. You centrifuge the blood. So if you have a blood here, inside this tube, these are the cells floating inside. When you apply the centrifuge, when you centrifuge the blood, you end up having what? You end up having the red blood cells collecting at the bottom, at the base of the test tube. Then you see some clear yellow liquid at the top. So this is what we call either the plasma. That is if it is found in the EDTA tube. So we call it in EDTA tube. It is called plasma. Then if it is in the if it is found in the serum separator tube, it becomes what? Serum separator tube. It becomes what? A serum. Okay, so let's continue. So we call it antiserum. The antibody molecules that bind to an antigen is commonly referred to as antiserum. Okay, the study of serum antibody and their reaction with antigen is called serology. So when you are doing without test, it's serology. Anything, when you are checking for hepatitis B virus, you use the plasma or the serum. It's serology. You are looking at antigen antibody reactions. That is direct enzyme link immunosorbent assay or direct ELISA. So it's what? Serology. Okay, so let's continue. So the blood contains plasma proteins. Everybody has plasma proteins in his blood. So of these plasma proteins, if you want to characterize them or measure them, we have different types of plasma proteins. We have the albumin and the globulins. And the globulins, we have alpha, beta, and gamma. But it is the gamma globulins that make up the antibodies. The experimental demonstration that experiment demonstrated that most antibodies are in the gamma globulin fraction of serum protein. So when you find serum protein or plasma proteins, the plasma protein binds to drugs, does a lot of things in the body. So if your liver is failing, you realize that you will start producing, your plasma protein level will be low. If you even take a drug, it will not work. So for the plasma proteins, we realize that albumin is here, but the gamma globulin is what we call the immunoglobulins, and they fight against infection. So if your plasma proteins are low, know that you are going to get a lot of infections because your gamma globulin concentration will be very low. And the gamma globulin is what makes up what? The immunoglobulin or the antibodies. Okay, so let's look at the antibody structure. Like you saw in the video, we have the variable chain, variable light chain, variable heavy chain. This all is heavy chain. From here to here too is heavy chain. So the heavy chains are 2. And the light chains are 2. This is a light chain. This is a light chain. That's why you are seeing Cl. Cl means constant. Constant light chain. Then constant light chain. Then variable light chain. Then this one is variable heavy chain, constant heavy chain, constant heavy chain, constant heavy chain. So the reason why this side and this side are variable is because their level expands. You know, they open their mouth to accommodate the antigen. They open their mouths to accommodate the antigen. So you can see this is the light chain, this is the heavy chain. So heavy chain is one, heavy chain another one, light chain here, light chain here. So this side will open its mouth to accommodate this antigen. So from here to here, from here to here is called variable because this is the antigen inside. This one too, antigen is here. So from you divide this into two, from here to here, you divide into 2. So the first part, that is from here to here, and from here to here is the variable chain, and from here to here is the light constant chain. And this all is the heavy chain. This is the constant heavy chain. This is the light chain. So let's go back to our slides. So we can see. So this is the structure, and they are joined together by what? Disulfide bond, sulfur-sulfur bond. So one may ask, what is an antibody? An antibody is a protein, as simple as that, and it belongs to a gamma. It belongs to a gamma globulin group of plasma proteins. So we have antigen. This is where the antigen binds. So we have antigen binding here. Antigen binds here, antigen binds here, like you saw in the video. And these one antibodies. Complement binds here, or immune complement binds here, but here antigen binds here, antigen binds here. So I've explained. So when we see CDRs, we have what? We have complementarity determining regions, or CDRs. Most of the differences among antibodies fall within areas of regions or variable regions called complementarity determining regions. So the variable regions, these VL and VL and VH are called the complementarity determining regions, or CDRs. And we have types of antibodies. We have the immunoglobulin M, immunoglobulin G. The immunoglobulin M is also referred to as mu. Immunoglobulin G is gamma. Immunoglobulin A is alpha. D is delta. Immunoglobulin E is epsilon. So each class can have either a heavy or a light chain that we know. Okay. So this is the structure. You see, I told you that this is the FC region. The FC region is from here to here. So all this is the tail is the FC region. It's called FC region because we will see that in an experiment that was conducted. So the FC region is called fragment crystallizable region. Fragment what? Crystallizable. Fragment. Sorry. So fragment crystallizable because it was treated with some chemicals and it crystallized. So we call it fragment crystallizable. And this is fragment antigen binding, the FAB. So all this is the fragment antigen binding. Look at it here. Fragment antigen binding. Look at it. So the same structure here. And Here it is being suspended by the plasma membrane, like we saw in the first picture that I drew on the naive B cell. Okay, so we have types, general structures of 5 major classes of antibodies. Okay, so for us to know that this is the heavy chains, the constant heavy chain, or these heavy chains is what we use to determine whether this is immunoglobulin A or B or C or whatever. The type of immunoglobulin. If you want to be able to memorize the immunoglobulins, you use a short abbreviation called what? Or mnemonic called GAMED. GAMED. GAMED means what? The G for immunoglobulin gene. The A for immunoglobulin A. The M for immunoglobulin M. And for E for immunoglobulin E. And D for immunoglobulin what? D. M. A. And G. And this is in a sequence in order of decreasing concentration in the blood or in the body. So you can see that immunoglobulin gene, this one, if it is in the blood, I'm just using my own mind. So if we have this one to be 50, this one will be what? 45. The A. M can be 30. E can be 25. And D can be 15. So it's in order or decreasing order of concentration. So when you look at the body, the highest concentration of immunoglobulin is immunoglobulin G, followed by A, followed by M, followed by E, and followed by D. The immunoglobulin G is what crosses the mother's placenta to go and fight infection in the fetus or in the developing baby. Then, or in the developing human being in the womb. Then the immunoglobulin A is found in breast milk, in vaginal secretions, in the intestine, gastrointestinal tract. Immunoglobulin M is what, if you have a new infection, that is what shows the immunoglobulin that you produce, and it is pentameric. It has what? 5 immunoglobulin joined together by a joining chain. Immunoglobulin E is what mediates allergic reactions or hypersensitivity reactions. It's the same as allergic reactions. The immunoglobulin D is the smallest immunoglobulin. Its function is not really known. Okay, so let's continue. So these are the types of immunoglobulin. We have the immunoglobulin G, which is the most abundant, following the gamete thing. Immunoglobulin G, immunoglobulin E, allergic reactions. Immunoglobulin A is a dimer. Dimer means 2, dimer means 2. It has dimerized. So there's a joining chain. The J means joining chain, joining. What is joining the two of them? Then this is a pentameric. That is what? A pentamer. That's the immunoglobulin M. So 5 different types, 5 immunoglobulin. The immunoglobulin M is 1. Then 5 of them joined together by a joining chain. So it is pentameric. Okay, so let's continue. So this is how, if you treat this immunoglobulin with chemical, this is what you get. So if you treat it with what? A pepsin, which is what? A digestive enzyme, you get the fragment crystallizable, this one, will be destroyed. So FC fragments are destroyed. So you have what? Fragment antigen binding alone. We have 2 of them. That's why we have FAB2. Okay, if you do, all these are digestive enzymes. So if you treat this normal antibody with papain, it's a digestive enzyme in the stomach. Hey, I don't know. It's not in the stomach. It's pepsin rather that is in the stomach. So this papain, if you treat it, this is some 2 scientists experiment, and they won an award in physiology and medicine in the past. They won Nobel Prize. So let's say, so this is fragment antibody fragment antibody binding, the fragment crystallizable. So when you treat with papain, this one, it breaks the hinge over here, breaks the bond over here. It breaks the hinge or the bond here, then we get 3 fragments, 2 of the fab like here. Then this one is not digested. Over here it is digested because you can't see it. Then when you treat with mercaptoethanol, which is a chemical, which is an alcohol, we have what? 2 heavy chains and 2 light chains. So this is an experiment. Antibody classes and biological activities. So immunoglobulin gene, let's start with the immunoglobulin gene. It constitutes about 80% of the total serum of immunoglobulin. I told you. And we have types: immunoglobulin gene 1, gene 3, gene 4. And they are those immunoglobulin that cross the placenta and play an important role in protecting the developing baby or fetus. So you read the rest. There are 3 types: gene 1, gene 2, gene 3, and what they do. Immunoglobulin E is the potent biological activity of immunoglobulin E, allowed it to be identified in serum despite its extremely low average serum concentration. So this is the last but one, or the second to last of the immunoglobulin. You see E. Look at the concentration. This concentration, I just put it there. That's not how it is in literature, but that's the penultimate, or the last but one, smallest immunoglobulin. The smallest is the D, followed by the smallest is the D, but the E comes before the D in abundance. So immunoglobulin E antibodies mediate, or they control, allergic reactions or hypersensitivity reactions that are responsible for the symptoms of hay fever, asthma, hives, and anaphylactic shock. The meaning of this is this. See, well, there is something we call some flowers or some green grass. When you come closer with the particles or the pollen grains that they produce, and you are asthmatic, it will give you what? Reaction. If you don't take care, you can't breathe. And you will be having nasal congestion, catarrh, and you will react. The solution is that you don't go closer to those things. But if you go closer, those reactions will happen. For instance, if you are asthma, you are not supposed to be using very, very strong sprays or even spray at all. Hives and anaphylactic shock, some of the time you can take in some drug or some food and it will give you reaction, to give you a whole lot of reactions. And the reaction can be what? It can even lead to you getting rashes. It can even lead to you getting rashes. It can even lead to you getting rashes. Let's see how an anaphylactic shock looks like. So if you get an anaphylactic shock, it is a medical emergency. Allergic reactions. It's a medical emergency. So look at him. He took some drug or some injection or even some food that his body does not like. Look at the rashes that he has gotten on his face. This man too is having a heart attack because of, and swollen eyes because of some medication or some food that he took. So those who want to become doctors, you have to be careful about the drug that you give to your patients. Sido and Co. You have to be careful. Sido and Annabelle and Abigail and Donko. You have to be careful. Prosper. Look, the drug that he took. Look at the reactions and the anaphylactic reactions. And it's medical emergency. If you don't take care, somebody can die because of this. So we have to be very, very careful. Very, very careful. The drugs should give people and others. Okay, so let's continue to our lecture. So immunoglobulin E, what it does is that it binds onto what? This is a mast cell. Mast cell is part of the cells in the body, and this cell, immunoglobulin E, which is an antibody, binds onto the surface of the mast cell. It will bind onto the surface with its fragment antigen binding section. Then so this is the mast cell with its nucleus. Immunoglobulin E will bind to it. When it binds to it, it will use the fragment crystallizable, this section, the FC section. This is the FC section. It will use it to bind to a complement. Alright? So when it binds it to a complement or any other thing, there will be degranulation. That means that the mast cell, the mast cell will now shed off its cytoplasm. It will secrete its cytoplasm, and it will release its there are many in the blood or in the system, in the body. So it will now shed off its cytoplasm, release its cytoplasm. So it's shedding off its cytoplasm. It will release all the granules in the cytoplasm. When these granules enter the blood, they are poisonous. It will end up giving you rashes, body itches, pains, swollen eyes. It can even give you tachycardia. Or tachycardia means what? Increased heart rate. Tachycardia. And bradycardia is what? Lower heart rate. Bradycardia is lower heart rate. So this will give you tachycardia. Tachycardia means what? Increased heart rate, or the heart will be beating so fast, like you are having BP and others. So your heart, your blood pressure will be high and your heart will be beating so fast. So if you don't take care and they don't admit you and stuff and give you, and most of the drug that they give over here, we call them, because these granules that the mast cell is produced because it has bound onto immunoglobulin E, the granules are called what? Histamines. And the histamines, when they enter the blood, trouble. You'll get rashes, you'll get heart attack, you'll get a whole lot of problems, headache and other things. So there are some drugs that we take that has been manufactured. If somebody has a medical emergency or hypersensitivity reaction or allergic reaction, we give him that drug. The drug is called antihistamines. Those will be going to do pharmacology. Dr. Akila will teach you some of these things, antihistamines. Let's continue. So that's, I've explained allergic reaction. That's what happens. There is what? Immunoglobulin E will come and bind. We use its fragment FAB section, or fragment antigen binding section, to bind onto the mast cell. Then it will also bind to complement and other cytokines. Then the mast cell will now, due to this, it will degranulate. It will release its granules. Degranulate. It will degranulate to release its granules into the blood, and the granules are called histamines, and that will cause rashes, body aches, body itches, increased heart rate, or pumping rate of the heart becomes so fast. And it's not bradycardia. Bradycardia is reduced or decreased heart rate. So the drug that you give is what? Antihistamines. This one. Antihistamine. Because we are producing histamine, so we have to give an antagonist of the histamine to stop the beating, to stop the histamine production. So immunoglobulin M, this is the first immunoglobulin expressed during B cell development. So naive or virgin B cells express monomeric immunoglobulin M. So upon maturation and antigen stimulation. So immunoglobulin M is the first immunoglobulin that the body produces. If you have during the primary response, the immunoglobulin that the body produces first is immunoglobulin M, and that's when your body does not know the organism. When your body does not know the organism is fighting with, first time you get COVID-19, first time you get any infection, the immunoglobulin that your body will produce is IgM. Okay, so let me explain what an antibody affinity and antibody avidity. You'll be meeting this throughout your career life. So affinity means what? How one of the a variable region binds onto an antigen. This black thing is the antigen, and the variable region is binding to it. Okay. Then avidity is when the two of them are binding the antigens on an organism. So the avidity is stronger than affinity. Affinity is only one paratope, or just one arm of the variable region binding to the antigen, whereas the avidity is both arms binding. So this side is binding, this side is binding. So we say that unity is what? Strength. If you do it alone, you don't get the results like if you do it collectively. So for instance, if you have this avidity and affinity, if you have this, so let's see, you have this organism, antigen, 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 antigen. This is a malaria parasite, antigen. So we have antigens, antibodies binding. Let me erase this. We have different antibodies binding. So we have this antibody binding to this. Yes. Antibody binding to this one too. Wow. Okay. Yeah. Okay, so we have this also. This also. And this also. Okay. So if only one antigen antibody binding is called affinity. The strength of binding for only one antigen antibody binding is called antibody. It's called antibody affinity. Then all of them, 1, 2, 3, 4, 5. So the bond between these antigens and this antibody, this antigen, this antibody, all of them put together gives you the antibody avidity. That is, the first one is for antibody affinity to this antigen. Then this one is what? The total strength of binding is called antibody avidity. So note it. Immunoglobulin M, I've explained what it does. That's the first immunoglobulin produced when you have a primary infection or primary response or first exposure to a particular infection. Immunoglobulin D is found at very low levels in the serum with a short, so when you use the gamete, that is D, GAMED, so that is D, so it is the smallest concentration. It has a short half-life which may be attributed to sensitivity of the molecule with the hinge region in particular The function of this immunoglobulin D is not clear, as it is not known to participate in the major antibody effector mechanisms, like I told you, so you can go and read on it. Immunoglobulin G is the predominant isotype found in the body. It has the longest serum half-life of all immunoglobulin isotypes. It is also the most extensively studied. So the gamete, this is gene, gamete. So gene, it has the highest concentration. So it has subclasses like gene 1, gene 2, gene 3, gene 4. So go and read what the classes do. It's very important. So go and read. Immunoglobulin A, serum levels tend to be higher than immunoglobulin M. For gamete, gene comes before what? The M. But considerably lower levels than IgG gene. Immunoglobulin A levels, this is what is found in the breast milk, in the salivary secretions. So the breast milk can contribute about 50% of the protein in cholesterol. It produces 50% of protein in cholesterol found in breast milk. Then the first milk given to the neonate by the mother, it contains immunoglobulin A. The breast milk contains immunoglobulin A, and the immunoglobulin A is also located at the mucosa, termed secretory immunoglobulin A. It's a dimer, like I showed you. It's a dimer. It contains two chains, so it's a dimer. You see there are two, 1, 2. So it's called dimer. It's called dimer. So let's look at. So it's a dimer. Okay. So these are the subclasses of the immunoglobulin A. These are the subclasses. So go and read on them. Immunoglobulin E, I've already explained. That is what binds to mast cells, and the mast cells will degranulate to produce histamines as a result of allergic reactions or anaphylactic shock. So after the degranulation producing of the histamine, the person can end up getting catarrh, rashes, headache, heart beating so fast, and the person can even collapse and faint or even die. And that is what we call anaphylaxis or anaphylactic shock. So it's very serious. If you know that perfume is not good for you, or excessive dust is not good for you, and you come closer with dust, and you tend to have just sweeping a room, you tend to have catarrh and your heart is beating so fast and you can't even breathe, your lungs are, then don't get closer to sweeping. Don't even sweep dust. Don't go closer to where there is dust or perfume, because there is no medicine for it. The only medicine is that don't go closer to these things. But another medicine is that they can give you antihistamines. Okay. They can also give you the antihistamines, and also we have other types of the antihistamines too. Okay, so let's go. So antigens and immunogens. So let's see. So macromolecules, generally proteins or factors that influence immunogenicity. Immunogenicity is what? The ability of an antigen to elicit immune response. So macromolecules generally either